here uh, we start looking at a very interesting system, which is carbonic acid. I want to look at some properties of carbonic acid, and to start with, the very special feature of carbonic acid is that it exists in two forms in water. One is carbonic acid itself, H2CO3, and another is CO2, carbon dioxide molecule dissolved in water. What's interesting that most of carbonic acid in usual aqueous solution is not in a form of acid but in form of dissolved CO2. Only small fraction, much less than 1%, is in form of carbonic acid. Now, carbon dioxide is in equilibrium with surrounding gaseous phase. So, when you have CO2 in gas and it is in equilibrium with water, this CO2 eventually will become partially carbonic acid, and this carbonic acid can dissociate to make hydrogen carbonate ion and H plus ion. When we make H plus ion, we reduce pH value and make our solution acidic. That's why acid. So, carbon dioxide, very stable molecule, double bonded here, is in equilibrium with aqueous solution, and constant of this equilibrium is named Henry constant, is around 30 atmospheres per mole per liter, which means when you have pressure of 30 atmospheres of CO2 above your water, you would have 1 molar solution of CO2 in water. In reality, we never have such high pressures, so we never have high molarities of CO2 in water. Next, a reaction of hydration of CO2 molecules, so making H2CO3 out of CO2 and water, is surprisingly very slow. Well, it's not very surprisingly. Look at this molecule, it's very symmetric, and now you need to bring one oxygen, completely changing the shape of the molecule, and for doing that you perhaps need to break some bonds here. So this reaction is very slow with half-life of 18 seconds. Opposite reaction is also not very fast. Half-life is 300ths of seconds, which is comparatively slow if you compare to usual protonation reactions that complete in microseconds, nanoseconds, and so on. This reaction of dissociation is fantastically quick. Now, the dissociation constant is relatively high, so our carbonic acid is relatively strong acid, but when we take into account that most of it is not in a form of H2CO3 but in form of CO2, the result is that acidity of CO2 solutions in water is not very high. Now, for life, this exchange is essential because we need to remove CO2 that is coming into our bloodstream from metabolic processes. And as you can see, it takes almost half a minute to make even half of CO2 in equilibrium. So, in living creatures and in us humans, there are special enzymes that facilitate this process. This is just a picture of human carbonic anhydrase enzyme. The working place is zinc atom surrounded by three nitrogens and some distant oxygen atoms. And all the process occurs somewhere here. This zinc atom is sitting inside enormous assembly of large protein molecules and plenty of these pinkish dots which are waters. Without this enzyme, you cannot be alive. You cannot breathe. Similar enzymes, but chemically different, are practically in all plants and in many other living creatures. So, let's look at equilibrium for CO2 in water in detail. We have first dissociation constant here, and we have second dissociation constant here, which makes carbonate ion CO3 2 minus out of hydrogen carbonate. Now, this second constant is irrelevant because we deal with acidic solutions, and this second process occurs only in strong bases, so we can skip it altogether. As we have seen, this Ka1 is a relatively big number. But because most of our H2CO3 is in fact CO2, this ratio is very small. You have a parent constant that is much smaller number, 4.5 10 to the negative 7 instead of 10 to the negative 4, and pK is 635 at 25 degrees. Now we can do all the calculations for equilibrium with gaseous phase if we'll take into account Henry constant and this distribution constant. We can calculate, I will not do these calculations, that our H plus will follow this equation. Now this again is a very small number, so we can simplify it even more for pH of carbonic acid solutions and equilibrium with natural air. pH will be 1/2 of log of this expression. I'm skipping this one. So hooking numbers for Henry constant, for equilibrium constant between two forms, and for acidity constant, and 1/2 of log of pressure of CO2 in atmospheres. So this number is 392 and minus log of PCO2 half. So if my pressure of CO2 is 1 atmosphere, log will be 0 and my pH will be 392, this number. With carbonated drink you may have higher concentrations of CO2, so your pH can go as low as 3.7. If you have natural concentration of CO2 in air is somewhere between 3.5, 4, 10 to the negative 4, your pH will be in vicinity of 5.6. So this is pH of water in contact with CO2 with no other solids or contamination in this water. In fact, there is some contamination and it brings pH of natural water slightly higher. Uh, at different concentrations of CO2, you have pHs in between, so between 4 and 5. Now, very important case is water containing hydrogen carbonate. And the most important example is plasma of our blood. So what happens in our blood? We have hydrogen carbonate and we have CO2 that is coming from our metabolism, and we want to remove it when we breathe. So the equation for pH is our well-known Henderson-Hasselbalch equation. pH is pK plus log of ratio hydrogen carbonate to CO2. Now, here it's a parent constant. We do not have gas inside our bloodstream. And value of this constant will be 635 at 25 degrees with no other ions. But this is not true for our plasma. We do not have 25 degrees first, and we have plenty of ions. So the real number is 6.1. Now pH of blood plasma is around 7.4, which when we solve this equation will give you ratio of 20. So 20 times larger concentration of hydrogen carbonate than CO2. Hydrogen carbonate is responsible for CO2 transport from cells through blood to your lungs. Part of this transport, however, is done by hemoglobin, same hemoglobin that is responsible for transport of oxygen. What's interesting that CO2 bonds not to iron atom of hemoglobin, but to free amino groups making carbonates, compounds of CO2 with amino group. This is the main buffer of our blood plasma and some of our tissues. Other buffers that are important in our body are phosphate buffer, hemoglobin, and proteins. In blood plasma, only carbonate buffer is dominant. In your urine, it will be phosphate and ammonia. In some other places, it will be proteins and ammonia and so on, so on. In general, metabolism produces more waste acids than it produces waste bases. So in urine, we have more acidity than basicity. Urine is acidic, and it is in part neutralized by ammonia, which is a base and which is excreted into urine as well. By changing acidity of urine, we can change balance of what's coming out of our body. Now, our normal pH is 7.4. What will happen if I have breath problems? For example, my lungs do not work well. I will have respiratory acidosis. Why? Because I have too much CO2 and pH will go down. It's not too much. If a pH is going down by more than 0.3, you'll be dead. But this is possible situation when you have respiratory problems. By measuring pH of your blood, uh, medical doctors can see what's going on with you. If they measure concentration of hydrogen carbonate at that time, they can figure out the reason of this. So there is respiratory acidosis, problem with breath, chronic problems with it. Can be metabolic acidosis. So something happens with your body, it produces too much acid. Now it can be opposite, respiratory alkalosis and metabolic alkalosis. All these can be taken into account, properly processed and addressed when patient is treated. The same situation, of course, occurs when patient is poisoned, and it's possible to figure out the problems with poisoning as well.